we would like to dive into the Moonshot Living Without Fear, and I'll give the word to Sander and Uli.
Yeah, thank you. I think Sander and I will switch back and forth in our presentation. So my name is Uli Scherer. I'm a rheumatologist at Leiden University Medical Center. And we felt that by discussing the Moonshot Living Without Fear, we should first focus on, with one slide, on what we actually mean with living without fear, what this moonshot actually entails. And I think that this in many ways refers to the problem that we have with many IMIDs, and that is the problem of chronicity. Because at the end, these are chronic diseases. They are characterized by either persistent or recurrent tissue inflammation. We, of course, see systemic inflammation, but each of these immune-mediated diseases has specific tissues which become inflamed. And what we see from our clinical experience is that what we can achieve at the moment is, in many cases, control of inflammation. So with many advanced therapeutics that we've that have become available over the years, we can quite well control inflammation. But this control of inflammation does not mean that the immunological activity of this disease is controlled or is actually stopped. So what we see is that we have a situation of clinical control, but immunologically, many of diseases are still active, and that's what we call immunological disease activity. So if you go one further, Tale, so because what that means for our patients is that we usually need to treat many of these patients for their lifetime, and that is especially in patients that are young, at young age, when they become ill, a big problem. And many of our treatments actually are also associated with long-term side effects. So, and that means that our patients not only need to be treated for a very long time, it also means that they are living with constant fear of flares or activity of their diseases. And some of these flares can be very, very abrupt, can be very unforeseeable, and that means living with fear. And where we want to go to with this moonshot is that they will have a situation of living without fear. So, with one further, we would like to focus on this immunological disease activity. And the problem is that for many of the diseases, we actually don't know exactly what immunological disease activity is. We don't have a clear definition, or, and that's very important, good biomarkers to actually monitor that. Because if you have a clear definition of what immunological disease activity is, you can also define what immunological remission is, so the absence of this activity, and you have a good biomarker, then you can start developing tailored treatments for these patients. And that brings us to the next point where we really want to go to, and that is cure, which means that we can eliminate the root cause of the disease, which keeps these diseases chronically active. And so there are a number of examples that we would like to present to you, steps that we are taking in this direction, and the first one will be presented by Sander.
Yeah, so thank you, Uli. I thought I will first provide a little bit more background, just to highlight what Tale also said, that in this immune-mediated inflammatory diseases, there's a lot of heterogeneity in the diseases between patients, which makes it often very difficult to predict treatment response, but also results in diagnostic challenges. And as you already said, Uli, there's a lack of good biomarkers to measure this immunological disease activity, which would ideally be present to guide our treatment decisions as clinicians. Because, of course, we want to provide personalized medicine for these patients and tailor treatment strategies, because not every patient is the same with the same diagnosis. So there's an urgent need for a new strategy to develop immune monitoring, both for patients and for clinicians. And in the next slide, I discussed the three phases of immune monitoring IMIDs. Of course, in the diagnosis process, there's an important role for immunological diseases activity and immune monitoring to guide your treatment decision. But then also the response to treatment should be monitored, as Uli said, because clinical control does not necessarily mean that there's also immunological control. And this is also important to guide decisions on perhaps even stopping treatment if a patient is in remission for a long term, to see if that is safe to do or not. So, and in the next slide, I just highlight our approach. So I think we need a combination of established and novel approaches to take us further. I think there's an important role for serology because that's very easy to obtain also in a home-based setting, for example. But also we should take advantage of the novel omics techniques that are available. And this could also be guided by more functional assays to see if we can determine if a disease is inactive or still active immunologically. And by application of these approaches across different IMiDs, we hope to develop a unified immune monitoring strategy that could help us in all phases of the patient journey, actually. And of course, we would like to design specific immune tests for certain diseases, but also preferably tests that could be used across multiple diseases to identify the immunological disease activity and also the type of immunological activity that a patient has to guide treatment decisions. So in the next slide, I just highlighted the long-term objective of this moonshot. So we would like to achieve real-time, minimally invasive immune monitoring, where patients are also empowered to take control of their own health. And of course, we are not there immediately. We need several quite big steps to get there. So we envision that by 2030, we will have immune profiles ready to drive future decision making in clinical practice. And then by 2035, we hope that we have implemented real world treatment tailoring based on the immune profiles. And hopefully in 2040, we will have this broadly implemented in at least 5 diseases, and also put patients at their own steering wheel to quickly evaluate whether they have an imminent relapse or not, for example. And this, of course, requires maybe the last part of the slide, if you go one back.
Right.
This of course requires a disease overarching collaborative research strategy, where we also need patient involvement, because it's important to at every step know how patients feel about this approach. And we need, of course, also valorization strategies to sustain this in the long term. And then the next slide. So I already highlighted the long-term objective, but there are some consequences if you want to achieve this. So we need actually preferably easy tests. If it can be based on serology, that would be nice, because you could, for example, have patients send in blood samples that they collect via finger prick test. I will give an example of that in a minute. But that also means that you should be transferring part of the healthcare to patient's home, for example. So that's more disruptive compared to how we are performing medicine at the moment. But I think it has a very good potential to essentially save healthcare costs. So in the long run, if we are able to do this, I think we will all benefit, not only patients and doctors, but also society. So in the next slide, I highlighted a recent example of how we approached the COVID-19 pandemic in the Target to Be consortium. At that time, of course, it started approximately 5 years ago. We were quite suddenly unpleasantly surprised by this pandemic, but that also required that the society was in lockdown. So it was very difficult to have patients come in to collect blood samples. So what we did was we reached out to patients via a finger prick test, so they could perform that at home and send it to us. And of course, we also performed some more cellular analysis, and for that, patients had to come to the hospital. But the majority of patients supplied finger prick tests to see if we could learn from this pandemic and how the body responded to the virus, but also what the impact of certain immunosuppressive drugs that our patients were treated with had on the course of the disease and the immunological consequences of being exposed to this virus. So in the next click, you will see that this is what is required for that. So a finger prick set that was sent to the patient. And of course, if you're talking about this large number of patients, and that's what we're talking about if you want to implement this in the Netherlands across all IMiDs, then you also need a different strategy. And in the next click, you will see that this requires teaming up with a mail order company, for example, to make sure that you receive enough finger prick tests and that that's also sent to the patients, etc. So it's a totally different approach than we usually take. So on the next slide, you'll see a different example of how this could be employed. So not focused on COVID, but on rheumatoid arthritis. This is a study from a couple of years ago, not done in the Netherlands, but in the US, where they took a radical approach of asking patients to weekly sample their own blood via finger prick test. They came into the clinic every month for disease activity assessments, but also some normal blood withdrawals to do cellular assays. And then also they reported their disease activity every week. What they next did was they performed RNA sequencing on the samples, and they also looked whether the clinical scores in the clinic correlated to the self-reported disease activity of patients, which was quite acceptable, not great, but quite acceptable. That's in figure B. And they could also demonstrate that the RNA profiles from the finger prick sets could be used to estimate the number of neutrophils, lymphocytes, and monocytes in blood. It correlated quite good with the PBMC samples that they collected as well. That's just the validation part of the study. And then in the next slide, the interesting thing came up. So they looked at disease activity over time and projected this as time to flare, which is in the first figure. The flares are indicated with a blue dot. And then what they did is they went to the RNA-seq profiles, and they could demonstrate that 1 or 2 weeks before flare, that there were changes in the profile of the blood. So in general, 2 weeks before onset of the flare, they identified a cluster of genes that were associated with a B cell signature. And then 1 week before flare, they could identify a fibroblast population in the blood. So that prompted them to come up with a hypothesis that there's already immune activation and changes in the immune system before you see the clinical onset of a flare. And of course, this requires a lot of sampling, but it's at least something that points towards the fact that the flare can potentially be predicted. So this is not recapitulated in other studies yet, and there's also some more other examples of maybe more B cell signature that Uli will discuss in a minute. But this is at least something that kind of shows that it is possible to predict the flare in rheumatoid arthritis by sampling at home, for example. So this could be a start of a project as well in the Netherlands to see if we can identify maybe the same signature or different signatures and see if we can set up such an approach in the Netherlands as well. In the next slide, I provide another example. This is a different disease, ANCA-associated vasculitis. It's a vasculitis of the small blood vessels that often affects the lungs and the kidneys, mainly in adults 50 to 60 years old. Men and women are equally affected, and it's characterized by autoantibodies against neutrophil antigens, most notably anti-PR3 and MPO. But in this managing this disease, it's often a challenge to find out if a patient presents with symptoms, whether it's AAV disease activity or an infectious complication of the disease, for example. So in the next slide, you can see that there's different ways to find out whether it's a disease activity or an infection. So you could look at ESR or CRP levels, so acute phase proteins, but that does not discriminate usually between disease activity and infectious complications. Some people closely monitor the ANCA titers in the blood, which is very much suited for diagnostics and is also regarded as a marker of disease activity, but it's not 100%. And literature is not really conclusive on this, so I think there are better markers required. And of course, you have other possibilities like the BFAS scale to see if the disease is active. But also some of these parameters are affected by infectious complications, so it's difficult to discriminate based on BFAS. Serum IgG4 has been used as a marker, but did not discriminate well enough. And then in Amsterdam, we figured maybe there's a different way of looking at this, and maybe we should look at the ratio between IgG4 and the IgG in the blood based on the RNA signal. So in the next slide, you will see that you can calculate the number of RNA molecules very easily from IgG4 or total IgG producing cells, and then you can calculate a ratio. It's a very easy qPCR test, and we could demonstrate that in active disease this ratio was really increased in patients. And if you use a cutoff of 11.2%, of course we need to validate this in bigger cohorts, but then you achieve a sensitivity of 86% and a specificity of 100%. So that is able to discriminate between active and remission. And we also have now some preliminary data that we're about to publish that this can also be used to discriminate between flare of the disease and an infectious complication. So that's something that we could also look into across different diseases and see if we could use it not only in vasculitis, but also in other IMiDs. And then I will hand over to Uli for example number 3.
Yeah, thank you, Sander. Yeah, this is an example in which we focused more on the cellular components of the immune response during different disease stages. And this is an example in which we looked at rheumatoid arthritis. So you have certainly followed the relatively recent developments in the field in which we see that B-cell depleting therapies are very successful in numerous autoimmune diseases. Which is one of the very good examples of a disease overarching effective therapy. But still, these diseases are, of course, different in their clinical presentation. And another thing that differentiates these diseases is their specificity in the immune response for certain autoantigens. So most of these diseases are characterized by autoantibodies, and they recognize different autoantigens. And many of these responses that generate autoantibodies are very specific for an individual disease. So we felt that it's important to look at this very specific immune response in individual diseases and look at the cells, at the B cells that make autoantibodies. So we developed a technology to identify autoreactive B cells, so B cells that make certain autoantibodies. And this is the example for rheumatoid arthritis, in which an immune response is generated against citrullinated proteins, and the autoantibodies are called ACPAs. We do that by using fluorescently labeled streptavidin molecules that are coupled to the antigen. And then we have the possibility to identify such reactivities in the peripheral blood of patients. And in the moment that we can look at these cells, we can understand their phenotype and whether they're activated or not. We can try to understand the biology, but also maybe identify biomarkers or targets that can be used to target such responses directly. And we do that in different disease stages that are depicted on the slide on the lower timeline. If you go one further, then there is an example on how such analyses can look like. So on the left-hand side, you see total B cells, different populations of B cells in patients with rheumatoid arthritis that are clustered based on their phenotype. So from naive B cells to memory B cells to activated memory B cells, and also plasmablasts, which are antibody-secreting cells. And so this is sort of the background population, and you can project on that B cells that are specific for certain antigens. So on the right-hand side, you see a similar picture for healthy donors. And in the blue cells, these are cells reactive against tetanus toxoid. So this is a response generated by vaccination. It's a protective immune response. And you see that these tetanus-specific cells, they cluster in a resting memory compartment mainly. But if you compare that to these ACPA reactive B cells, then that's the next slide. Then these are depicted in orange, and you see that these cells are very activated, and they generate a lot of plasmablasts that make autoantibodies. If you click one further, then you can study their phenotype in detail. You can look at markers that they express, and what you basically see are many activation markers. These cells proliferate very actively, and they belong to a certain activated memory B cell subset. So if we go one further and we put that into perspective of the disease, then if we take a look at this lower timeline, this is the time sort of disease trajectory that patients usually go through when they develop this disease and have the disease. So from a healthy situation, to a situation which we call at risk, in which there are antibodies present but not a disease yet. And then there's a phase which we call clinically suspect arthralgia. So that's a phase in which patients have signs of autoimmunity, such as autoantibodies, but no clinical signs of the disease yet in the sense of arthritis. And then we have the onset of disease and, of course, the established phase. In some patients, not many, they reach, after having been treated, a phase of drug-free remission, which is sustained. So they remain in this remission phase for a long time. And so if you study the phenotype of such autoreactive B cells in these different stages and specifically focus on this pre-phase clinically suspect arthralgia and the phase of the individuals that reach remission without having to use any drugs anymore, then you see, and that's depicted on the left-hand side as with one activation marker as an example, that individuals who have clinically suspect arthralgia, who do not develop arthritis over time, they show a certain expression of this marker. But the individuals who will develop rheumatoid arthritis, their B cells, they are as active as the ones in established disease already. So we actually see the immune activation before the patient develops the full-blown picture of the disease. And if you compare that to individuals who reach sustained drug-free remission, then you see that the phenotype of these autoreactive B cells becomes more similar to those individuals who are in the pre-phase and who do not or maybe do never develop disease. And a similar example is depicted on the right-hand side. So this is the frequency of plasmablasts, in which we also see that the phenotype of individuals who are in this arthralgia phase and do not progress to rheumatoid arthritis is very similar to the individuals who are in drug-free remission. So this looks as if the immunological profile or degree of activation tells you something about the trajectory that the patient goes through, but it also shows you what immunological remission could look like on an immunological basis. And if you go one further, this is more a list of things that you conclude. We saw that this activated phenotype that I showed you persists in individuals who have rheumatoid arthritis and who reach clinical remission, which we induce by our current treatments, so anti-inflammatory treatments such as TNF inhibitors or JAK inhibitors. So there we see clinical remission, but no immunological remission. If we use a treatment such as abatacept, which inhibits the stimulation of B cells through breaking the co-stimulation with T cells, then we see that we can actually silence this activated phenotype. This is transient as long as we give the treatment. If we remove it, then the activation comes back. But it's very interesting to see that other groups have shown that if you use abatacept in the pre-disease phase and you sort of inhibit this B cell activation, you can actually delay in certain individuals, even prevent the development of rheumatoid arthritis. So this translates this observation directly to clinical impact. We are currently studying autoreactive B cells in other autoimmune diseases. We see similar activation stages. This is very important to link it also to vaccine responses or to responses against antigens induced by, for example, COVID, which is done extensively at St. Queen in Amsterdam. And it allows us to understand what immunological remission actually looks like. We see similar findings in other autoimmune diseases, and also here you can sort of try to understand what is immunologically really happening. And this brings us back to this idea that we want to induce immunological remission, which at the end comes closest to what we think is a cure of an otherwise chronic disease. I think that with this, I hand it back to Tale.
Yes, thank you very much, Sander and Oli. Very nice overview of current technologies and how these potentially can be translated across diseases. So I think it's good to briefly stand still on the opportunities for collaboration. So what's in it for you is always an important question. So why would you be interested in ImmuneHealthXL and in the hopefully upcoming PPP projects? Well, by joining up with ImmuneHealthXL, you have the opportunity to collaborate with a national network of immunological experts across IMID. So this is not only on the research side, but also on the clinical side. And I think it's interesting and important to realize that you build upon years of expertise, technologies, and infrastructures which have been built up in these previous projects, and it's still available within this network and accessible in that sense as well. For a small medium enterprise or spin-offs, this is a great opportunity, I think, to be able to establish proof of concept for your technology or for your potential drug candidate, and you can do this across multiple diseases. And of course, this allows for funding opportunities. This Health Holland public-private program is one example, but we are continuously looking for other funding opportunities in general. So the next steps, and this is really on the funding side, is that we have submitted this application. We are quite optimistic about the outcome, but of course we have to wait till October to hear from the committee if this is awarded. And then in January, we open a call for invited pre-submissions. So if you think you've got an interesting technology or an interesting project idea, please do reach out to us or to Sander or Uli, and we can link you to potential collaborators within the ImmuneHealthXL network. As I already mentioned previously, we also are always on the lookout for other funding opportunities. And we would like to collaborate with you as well. So these can be NWO consortia, Horizon Europe projects, or maybe bilateral multi-party public-private collaborations. And if needed, we can support in helping these projects get together or facilitate in the networking. So do feel free to reach out to us. And having said that, I'll